Hello students, welcome to the class today. So we are going to the next class of our CBSE NEET journey, and that is biotechnology, the principles and processes. So in simple words, we could say that biotechnology is a branch of science that uses living organisms, that is biology. Bio refers to life or living organisms or their parts like cell or enzymes to create products and solve problems. We are using technology in living organism to produce some products. It could be something like enzymes, or like synthetically we are producing some enzymes, or we are solving some problems, or we are creating commercially producing some products, etc. So it involves using biological systems. That is why it refers to biotechnology, like bacteria, plants, animals, and they are used in the industries such as medicine, agriculture, environmental management to improve our lives. So we are using technology in living organism to improve our life. For example, in medicine, we are able to produce, for example, like insulin for diabetes. We are able to create vaccines, as well as recently we are being in a stage where we have developed gene therapies, as well as we are able to cure the genetic diseases as well. Now moving to agriculture. So this will be included in the next chapter. Tomorrow we'll be having the class regarding this agriculture, that is creating drought resistant crops or pest resistant crops like that. Now in environment also we do have the role like cleaning up oil spills or waste management, what we learned yesterday, reducing pollution using microorganisms, etc. So, in medicine, biotechnology helps in creating medicines, vaccines, and therapies. In agriculture, it's used to create genetically modified crops that can resist diseases or grow better in different conditions. And in the environment, biotechnology helps to clean up pollution or recycle waste. So that is what biotechnology is. And in this class, we are going to learn about the principle and the processes which we are conducting during this technology. Actually, the history of biotechnology stretches back thousands of years, even before the term biotechnology was coined. That is ancient times, the human have been using simple biotechnological process for centuries, even before the science was understood. That one is fermentation. Yesterday we learned it, right? In detail, we learned it about fermentation to making bread, beer, and wine. Unknowingly, they used microorganisms to convert sugar into alcohol and carbon dioxide. So actually, it is included in the microbes as microbes for human welfare chapter also. And another term which comes under this topic is genetic engineering. So in 1970s, the scientists began to experiment with genetic engineering, where they started modifying the DNA of organisms to achieve desired traits, like producing human insulin from bacteria, many vaccines like that. Okay, so now we are moving to the principles of biotechnology, and we are moving to learn the term genetic engineering. So we are doing some engineering works in the gene. So we actually were, before two classes, we were discussing about the molecular basis of inheritance. And in this chapter, sorry, inheritance, in this chapter, we discussed about the DNA, and the functional unit of DNA is gene. Now we are going to learn the technology we are using on this gene to create a desired effect or desired product. Now, genetic engineering. These are like the term itself, like the technology. These are the techniques to alter the chemistry of genetic material. So if we are having a special or a significant type of DNA in our cells and we want to alter them, we have to use some kind of techniques or instrumentation or methods. So that is what genetic engineering is. And this DNA, this altered DNA is introduced into a host organism like bacteria, and it changed the phenotype of the host organism. So when this particular gene of interest or the desired gene is taken or transformed to a particular organism, its phenotype will be changing. Understood? So we are inserting a desired gene or gene of interest. We use different terms. The terms are desired gene or gene of interest. So these are the terms. So another definition is genetic engineering is in vitro. That is very important. It's not an in vivo process. It is in vitro manipulation of genes to get desired product or character. Simple definition. Genetic engineering is in vitro manipulation of genes. They are manipulating, they are changing, they are altering, manually they are doing it. Okay? To get a desired product or character. That means a phenotype. So we have to maintain a sterile ambience for this process. So in chemical engineering process, to enable the growth of only the desired microbes, that is eukaryotic cell, in large quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes, etc. Okay, so we have to maintain a sterile ambience because we are working with some organisms. So we need the microorganisms or eukaryotic cells in a larger quantity, and the products, like we were discussing about the desired products here, right? And that include antibiotics, vaccines, enzymes, etc. Now, moving to the next slide, the techniques of genetic engineering. So now how it is being carried out in the laboratories. So it includes the creation of recombinant DNA. It is also mentioned like rDNA, that is recombinant DNA. R stands for recombinant. Recombinant in the sense we are combining two kind of DNA. For example, if this is the normal DNA of an organism, as we discussed in the previous slide, it is the bacteria, that is a host DNA, and we are inserting a particular DNA to it, that is the DNA, the desired DNA or the gene of interest. So here we have the recombinant DNA, rDNA. Understood? So it is a recombinant. That means we are combining two kinds of DNA. Now, use of gene cloning and gene transfer. So this is another method where we use the technique of genetic engineering. That is, we use gene cloning and gene transfer. Gene cloning in the sense if we are having particular kind of DNA, that is gene, and we are transferring to a host cell and we are making more copies of this particular DNA, that is known as gene cloning. And the gene transfer is transferring of that particular DNA to another organism. Okay, now, yeah. So this helps to overcome the limitation of undesired genes getting multiplied along with the desired genes in traditional hybridization technique. So here, as we discussed, like we are creating recombinant DNA, we are doing gene cloning. That means we are making more and more this recombinant DNA here, more and more recombinant DNA. That is gene cloning. We are copying the similar kind of DNA and we are transferring this DNA as well. So this is helping to overcome the limitation of undesired genes getting multiplied along with the desired genes in traditional hybridization technique. So in traditional hybridization technique, for example, if we have both desired and undesired DNA, so there is no way that we can avoid it. Both the genes will get expressed there. But by the means of genetic engineering, we can overcome this limitation. That is, we can stop the multiplication of undesired gene. We can remove them. We can have the desired gene there. Okay. Then it also allow us to isolate and introduce only one or a set of desirable genes without introducing undesirable genes into the target organism. So this allows us to isolate. We are isolating a particular DNA and then we are introducing only a one or a set of desirable genes without introducing the undesirable, the unwanted genes into the target organism. Now, in a chromosome, there is a specific DNA sequence which is called the origin of replication. So we have gone through this term in our previous chapter, that is molecular basis of inheritance in replication, the origin of replication, where the replication is initiated. So this is a particular site or the specific site in DNA or gene where the replication is initiated. So this is a mandatory site in gene for the DNA replication. So as similar to in case of DNA, recombinant DNA technology also, we need this origin of replication. Now, so for the multiplication of any alien piece of DNA in an organism, it need to be a part of a chromosome which has a specific sequence known as origin of replication. So that means the ori site should be accompanied by the gene of interest. So if this is the gene of interest, then this should be accompanied by the gene. This is the gene. This should have an ori site preceding it so that it can like carry out replication, that is replicate. Okay, that means it can create a clone, it can create a copy of itself. So an alien DNA is linked with the origin of replication so that this alien piece of DNA can replicate and multiply itself in the host organism. And this is known as cloning. We already discussed the term, right? Cloning. Now this diagram well explains the recombinant DNA technology or genetic engineering, where we are considering this as the particular gene of interest. Okay. Let me take another ink here. Yeah. So this is the gene of interest which we want to isolate it and we want to make more clones and we'll be like using it for our interest, like it might be like commercial production of something or we want to transform some animal like that. So this is a gene of interest. Now we need a vehicle, a transporter to be like so that we can be transporting this particular gene of interest to the bacteria, which is the host cell. Okay, so we need another, that is a vector. The vehicle is called the vector, cloning vector, and that is the plasmid. Plasmid is an extra chromosomal DNA. Other than the nuclear chromosome, the bacteria are having an extra chromosomal DNA called the plasmid, which is circular DNA, and we insert the gene of interest into this plasmid and we form the recombinant plasmid. And then we insert this recombinant plasmid to the bacterial cell. And this process is called transformation. Okay, so as a result of transformation, what happened? We inserted the gene of interest, that is combined with the plasmid, the recombinant plasmid into the host cell. And later on, this will be combining or it will integrated into the host genome. It will get integrated into the host genome and will get an recombinant DNA, rDNA. Okay. So what are the basic steps involved in this process? First one is isolating genomic DNA. So if you need to take this gene of interest from human DNA, you have to isolate the DNA because we know that we have the cells, tissues, samples, whatever we are collecting, and it will be consisting of all the biomolecules together. It might have carbohydrates, enzymes, proteins, amino acids, so many kind of components or molecules are present in our body. So we have to separate the DNA, the genomic DNA from them. It is isolating the DNA from them. Okay. Now, second is fragmenting the DNA. So once we got the DNA, and if this is the particular gene of interest, now what we have to do is that we have to separate this particular DNA from the sample. That is, we have to fragment the DNA, fragmenting, separating or cutting the DNA using molecular scissors. That is restriction, that is restriction into nucleases. That is what mentioned here, REN. So you can like compare this diagram here. It is not exactly like this. They are using enzymes here, but that is why it is molecular scissors. It is acting like a scissors. It is working, or the principle behind is like a scissor, but it is a molecular scissor, not the exact physical scissors. So this particular enzyme can cut the DNA. That is fragmenting. The term is fragmenting. Now, the third step, screening the fragments for a desired gene. Now, what we saw is that we have so many fragments here. First is the gene of interest we have, and then we have so many small other fragments, isn't it? So we have to find out the one which we need. It is a gene of interest. We have to identify the one. That is, screening has to be done for the fragment of the desired gene. We have to remove all the unwanted fragments and we have to isolate the desired gene. Now then you have to insert the fragment with the desired gene of cloning vector. That means a plasmid. So you will be taking a plasmid, you will be cutting the plasmid also by the molecular scissors or restriction into nucleases, and then you will be inserting this gene of interest into this plasmid. Okay. Now, once you are ready with the recombinant plasmid, so this is the recombinant plasmid, our plasmid, that is recombinant plasmid, which is having plasmid plus the gene of interest. Now, next is introducing it into the host. So, introducing the recombinant vector or plasmid into a competent host cell. Competent host cell, that means which can survive in any kind of situations, which can accept this new DNA, which can accept the gene of interest and incorporate that gene of interest into its own DNA. That is a competent host cell. So we will be introducing the recombinant vector into the host cell. Then we'll be culturing the cells. We'll be culturing the, if it is bacterial cells, to obtain multiple copies or clones of the desired DNA fragments. So that we'll be getting more and more copies of desired genes. Okay. Then next is using these copies to transform suitable host cells so as to express the desired gene. Actually, this particular host is like a starter or an initiator so that we can get so many clones. So then after getting more clones, we will use these copies to transform the suitable host, the actual host, whatever we are working on. So we'll be using the suitable host cells to express the desired gene. That is called transformation. Okay, so these are the basic steps involved in genetic engineering or recombinant DNA technology. Now, as we discussed the plasmids, so plasmids are, as I mentioned, they're extrachromosomal DNA. So this is a plasmid of Salmonella typhimurium, which is a vector plasmid, and we'll be taking this and cloning it in the E. coli. Okay, so they took this plasmid from Salmonella typhimurium and they added the gene coding for the antibiotic resistance it with, and then it is cloned, it will be cloning it in the E. coli. So recombinant DNA technology, also called genetic engineering, both are similar terms, deals about the production of new combinations, that is recombinant, new combinations of genetic material, that is genetic engineering, genetic material artificially in the laboratory, that is in vitro. It refers to in vitro, artificially in the laboratory. And recombinant DNA, that is rDNA molecules, are then introduced into the host cells where they can be propagated and multiplied. Okay. Now, moving to the next part. So, this is an example of a recombinant DNA technology where human insulin is produced. So, this is a human cell with the gene of interest, that is the insulin gene in DNA, the insulin gene. And we are inserting this insulin gene into the DNA of the bacterium. So, this is the bacterial cell without the insulin gene in DNA, and the insulin gene is inserted in the DNA of the bacterium. So here we have the recombinant DNA. And now this is inserted into a host cell, so that bacteria is multiplied where we have more clones or more copies of the DNA, and ultimately what happens, they will be producing the human insulin. Okay, the similar one is shown here also. Now, so basic steps of rDNA technology, the identification of DNA with desirable genes. So you have to first of all identify the DNA with desirable gene. That means, for example, like right now we discussed about insulin. Okay, so insulin is produced by the pancreas. So pancreas will be having this particular kind of DNA which is active in pancreas, and they will be converting this DNA to particular protein that is called the insulin protein. So you have to identify which DNA is causing the production of the particular protein, that is insulin. And then you have to introduce the identified DNA into the host. And also you have to maintain of the introduced DNA in the host and transfer of the DNA to its progeny. Now, the tools of recombinant DNA technology. Now we are moving to the tools required for recombinant DNA technology. First is enzymes. We need, as these are chemical reactions, we need enzymes for this reaction. And the main enzyme required is, as we discussed, the molecular scissors. What is it? Restriction endonucleases. And it cuts the DNA at specific sites. We have already discussed it. Second enzyme is DNA ligase. And this can be connected to the term ligation. That means joining. Okay, helps to join two DNA molecules. Now the second tool is the cloning vector. We already discussed that is a plasmid. It's also known as the vehicles for cloning, the cloning vectors. So vectors serve as a vehicle to carry a foreign DNA sequence into a given host cell. For example, plasmids, cosmids, or bacteriophages. Then, third tool we require is the host cell. For example, the bacteria. And in the bacteria itself, what can be, usually we do it in the E. coli. So the cell into which the rDNA is transferred for multiplication of desired gene copies is usually E. coli, that is a host cell. Finally, we need a bioreactor. That means, if, for example, if we want to produce insulin in a larger quantity, we require a instrumentation. For that, we can culture the bacteria and improve or enhance their growth, multiplication, and the expression of the genes, so that we can produce more and more insulin. For example, I'm considering like insulin production. So specially designed container, actually what is bioreactors? They are specially designed containers which is used to carry out the biological reactions under controlled conditions. What is controlled conditions refers to? It is like the temperature is maintained, the pH is maintained, so that the growth of the bacteria can be increased, but still it is controlled, and removing the waste, adding the raw material, all are regulated manner. Okay. So now starting with rustication enzymes, we discussed it as molecular scissors. They belong to a larger class of enzymes called nucleases. So nucleases are the enzymes which cleave the nucleic acid. And what are the two types of nucleic acid? DNA and RNA. So the enzymes nuclease cleave DNA and RNA. And there are two types of nucleases, exonucleases and endonucleases. So what they do is that they remove the nucleotides from the ends of the DNA. That means they'll be not from the ends of the DNA means that is done by exo, and the nucleases which cuts at the specific position within the DNA is endonucleases. For example, if this is a DNA, if the restriction enzymes are cutting from the end, then that is exonucleases, removing the nucleotides from the end of the DNA. Now, if they are cutting, like specific cuts are made at particular positions, they are cutting within the DNA, we are removing this particular site from DNA, that is endonucleases. Okay. Now, coming to examples. The first restriction into nuclease, that is hint2, always cut the DNA molecule at a particular point by recognizing a specific sequence of 6 base pairs. Remember this? 6 base pair sequence that is recognized by the hint3 enzyme. So hint3 is the restriction enzyme, restriction into nuclease enzyme, REN. Okay, so that is the example is hint2. They cut the DNA molecule, they'll be cutting the DNA molecule at a particular point, and that is a particular sequence as well. And this sequence consists of 6 base pairs. Clear? And this specific base sequence is known as the recognition sequence. So what is this base sequence known as? Recognition sequence. If they find out that, if the enzyme find out that if this is the desired recognition sequence, they will cut, like they'll be cutting at this particular site. So each restriction into nuclease recognizes a specific sequence in DNA, that is the recognition sequence, and that is palindromic nucleotide sequence. So that sequence are palindromic nucleotide sequence. You know what is palindromic? For example, Malayalam. So this is this is a language spoken by a particular group of people in Kerala, not particular group of group of people in India, that is from Kerala. And the term, that is Malayalam, if you read from left to right or right to left, it is similar. Malayam, similar here also Malayam, isn't it? Another term we could say that what can be let me think. Yeah. Um, can you think about any kind of term? Let me think also. Rougher, isn't it? Refer. Yeah, this is a palindrome. We can also suggest any kind of palindrome sequence from REFER or RE, like both sides it is similar. There are so many palindrome sequence like terms. Then what? Yes, exactly. There are so many. So these are the group of letters that form the same words when read both forward and backward. And here this recognition sequence are palindromic in DNA sequence. This is palindrome in DNA. That is a sequence of base pairs. It reads same on the two strands when orientation of reading is kept the same. So here we can see the sequence here. This, yeah, TTAGCAC. Here also TTAGCAC. See, this is palindromic. And here again GTGCTAA. And here AATCGTG. So if you are reading from the sequence, like if you are reading from here left to right, and if you are reading from right to left, it will be similar. Okay. Now, so now how they are going to cut at this particular site. So the enzyme cuts both DNA strands at the same site. So this particular enzyme will be cutting the both DNA strands at the same site. It will be cutting at the same site. They will be cutting, and this is a vector DNA, that is the plasmid, the vehicle. Okay, so here we have the palindromic sequence GAATTC, and here also GAATTC. That is a palindromic sequence. It is not like the GAATTC here, also not like that. This sequence is palindrome to the opposite sequence. Okay. Now, EcoR1, that is another example of endonucleases, restriction endonucleases enzyme, RN. It cuts the DNA between the bases G and A. So they are really specific. It is very important to understand. They are recognition sequences, and these enzymes are really specific. So they will be cleaving the DNA or cutting the DNA at particular site between G and A. Here, G and A. Only when the sequence GAATTC is present in the DNA. And the sequence, what is the sequence? You have to by heart the sequence as well. GAATTC. GAATTC. There is 6 base pairs. So here you can see they are cleaving at G, between G and A here, and here also between G and A, and you got a sticky end. This is a sticky end, like that you can, yeah, this is the sticky end. So the other side will be like this, so that you can join it together. Okay, so see, it is exactly complementary here. DNA fragments join at the sticky end. So here we have the recombinant DNA. Understood by this diagram? It is easy, right? Now, so just now we learned about the term sticky end, restriction endonucleases. Now here we are having sticky end in the sense they are like compactable with each other. So they'll be like, it just like a lock and key. They are just exactly fixing together. And here it is a blunt end. Blunt end in the sense they are exactly cutting. No overhangings are there. Here they are complementary. Overhangs are there. Overhangings, isn't it? Here there are no overhangings. They are uniform edges. Uniform edges. So here they are cutting in between G and A here. And yeah, that is a cleaving happening here. So that is a blunt end restriction endonucleases. So here we were discussing about EcoR1, which cleaves at GAATTC, and they particularly cleaves at G and A. Okay. Now, moving to next part. Yes, this is similar. So here we have the foreign DNA or the restriction enzyme. We are cleaving and we are taking the particular gene of interest, and we are inserting the gene to the vector DNA or plasmid at particular site, and that is done by the ligase enzyme. Then transformation, the E. coli will be receiving the recombinant DNA, and then the cells divides. That means what will happen? The cloning of the DNA happens, cloning the DNA, and finally we'll produce the product, the desired product. So this is a sticky end. Yeah, actually here the animation is not working. Yeah, we'll move to the next slide. Now we are moving to naming the restriction endonuclease enzyme, RN. We are going to name how we are not going to name how we are like in what basis we are naming the restriction endonuclease. Right now we learn 2 types or 2 examples of restriction enzymes, EcoR1 and HIND3. So let's go to the next slide first. So EcoR1, E refers to Escherichia. Okay, and co refers to coli. That is E. coli. So EcoR1 is taken or extracted or isolated from E. coli. So that is Escherichia coli. And R refers to strain by RY13. And this is Roman letter 1, which is depicting the first endonuclease to be discovered, the order of discovery. Similarly, HIM3, H stands for Haemophilus, IN stands for influenzae, and D stands for the strain RD, and 3, the Roman letter, is the third endonuclease to be discovered. So that means what we can conclude, the first letter of the name comes from the genus. For example, Escherichia, Haemophilus. And the second two letters come from the species name. E. coli, CO, right? And here IN, influenzae. Now, the fourth letter is in the capital, capital is in capital. So, echo R, capital R is there, hin. So, this is fourth letter is in capital, derived from the strain of the microbe, which strain it is from. And the Roman letter followed is the order of the discovery. On what order? It is like what order in the missing, what is the number of discovery. Okay. Now, moving to the next slide. So, separation and isolation of DNA fragments. That means the DNA of the interest, the gene of interest has to be like separated. So, cutting of DNA by restriction into nucleus results in the fragments of DNA. We discussed it earlier, right? Now, these fragments can be separated by a technique known as gel electrophoresis. Okay, and the DNA fragments are separated according to their size. So based on the size, they are separated. Now, what is the technique used? Gel electrophoresis. So what is electrophoresis? It is a technique where we use the electricity, the current, to separate a particular biomolecule. So it is separation is based on the charge, like what charge they are possessing, the size of the molecule. As coming to charge, as DNA is negative charge, they will be moving towards the what? It is an anion, negative charge. Anion will be moving to the cathode part, isn't it? So that will happen in a uniform way, and based on size, we can separate the DNA fragments because each DNA fragments will be of different sizes. Here you can see they're separating. See, these are the differences of sizes. Due to the difference of sizes, they are separated. So the separated DNA fragments are visualized. Now how we are going to identify them? We are visualizing by staining the DNA with ethidium bromide followed by the exposure to UV radiation. So once we isolate the DNA and we separate the particular DNA fragment, that means we are isolating the gene of interest, then what we'll do that we'll run the gel to separate the particular DNA. So we will be adding a dye, that is ethidium bromide. For example, I'm just to understand, I'm showing it. So we'll be adding this ethidium bromide and then we'll be running the gel, and after we'll be using the UV illuminator, we'll be under UV watching this particular DNA under the UV light, and we can see what color band is there. Bright orange colored bands will be appearing. So the separated bands of DNA are cut out from the agarose gel. This is agarose gel, and extracted from the gel piece, and this is known as elution. So if you find out that this particular one is the gene of interest, then what we will do? We'll cut this gel part and then we'll be extracting that DNA from that area, and that is known as elution. Okay, like a purification we can see. And these DNA fragments are purified and used in constructing the recombinant DNA with cloning vector. Now, here you can see this is showing a bacterial chromosome here, and this is the plasmid having the gene of interest. So plasmids are small extra chromosomal double-stranded circular forms of DNA that replicate autonomously. So this is also another extra point. They replicate autonomously. That means they replicate themselves separately. They are replicating. So they are having their own machineries of replication. That is what I mentioned. So they are vehicles of cloning. Now the salient features of a vector. So it should contain an origin of replication. We started with it, right? They should have an origin of replication so that it is able to multiply within the host cell. They have to produce more clones. That means they have to replicate. So they need the oricide, that is origin of replication. Then it should incorporate a selectable marker. So what is a selectable marker? So you might have seen like you are having particular uniform, like a specific uniform for a particular school, and even in case of different schools will be having different uniforms, or in case of like considering a police department, they will be having, we are having different offices, like different departments even in the police organization. So like for the group of police which are working under the traffic will be having a different one, or even in the army, the navy are having different uniforms. So why we are having different uniforms? To identify them, to select, to recognize them from a group of people. In the same way, if we want to find out, like, even is this our recombinant, the host carrying the recombinant DNA or what? So in order to find out that, what we have to do, we have to add, like they should incorporate a selectable marker. For example, the ampicillin resistant gene, that is antibiotic resistant gene. In this diagram, it is ampicillin resistant gene or tetracycline resistant gene, that is antibiotic resistant gene. That means this, the bacteria which is having this particular gene will be resistant to the particular antibiotic. That is ampicillin, tetracycline. These are the antibiotics. And if the bacteria is having this ampicillin resistant gene, then that means this bacteria can survive a media containing the antibiotics, so that it can survive over there, and we can identify whether it is the host organism carrying the recombinant DNA or not. Okay, it is a selectable marker, so we need a marker DNA as well. Now, they allow to select the host cells that contain the vector from among those which do not. The vector must also have at least one unique restriction into nuclease recognition site. That means the site where the DNA has to be inserted. So the vector, we know that already we are cleaving at two particular sites, isn't it, for the insertion of DNA. So in order to insert a gene of interest, we have to cleave or cut the plasmid as well. So that means they should have a unique restriction into nuclease recognition site, the palindromic sequences, right? So the vector should be relatively smaller inside. They should be small as well. And the most commonly used vectors are plasmids and bacteriophages. The most commonly used vectors are plasmids and bacteriophages. The TI plasmids contain the tDNA, and retrovirus can be used. So TI plasmids are, we are going to learn about it later. And the retrovirus is the RNA of like the viruses do have, some viruses do have RNA as their genetic material, and that is the retrovirus. So the TA plasmids which have the tDNA and the retroviruses can be used as the vectors. So this is the examples of cloning vectors, PUZ19. Here you can see that they are having ampicillin resistant gene, that is a selectable marker, the ORI site, the promoter sequence, and these are the restriction sites, ECO-R1, HIN3, the multiple cloning sites. Now, LAG-Z is the gene for beta-galactosidase enzyme in E. coli. Now here, this is another example, PBR322, a vector. It also has the origin of DNA replication, the ORI site, then the ampicillin resistant gene, the selectable marker, the ECO-R1, HIN3, BAMH1, SAL1, CLAV1. These are the restriction sites. It also has tetracycline resistant gene as well. Now, moving to the identification of recombinants, how we can find out or identify whether the organisms grown here are in the culture are recombinant or not. The most efficient method of screening for the presence of recombinant plasmid is based on the principle that the cloned DNA fragment disrupts the coding sequence of a gene. So if a plasmid is having a particular DNA fragment, this DNA will disrupt the coding sequence of the gene. That means if there was a beta gal gene in the plasmid and we inserted a gene of interest, and that means the gene of interest is added here. So what will happen? The activity, the expression of this particular gene will get interrupted, and that is called insertional inactivation. So if this particular gene is expressing, that means this is not successful transformation. The gene of interest is not completely inserted into the plasmid. If this gene is not expressing, that means this particular beta gal gene which was originally present on this plasmid got interrupted. That is insertional inactivation. They got inactivated. This means they are not expressing. So the powerful method for screening for the presence of recombinant plasmid is referred as blue-white selection. So what is blue and white selection? So this is based upon the insertional activation of the LAC-Z gene present on the vector. So we just find it out like LAC-Z gene present on the PUZ19 vector. So actually the LAC-Z gene codes for the enzyme beta-galactosidase, which can cleave a chromogenic substrate into a blue colored product. That means here the substrate can be converted into a product which is blue colored product. Okay, so this reaction is catalyzed by the enzyme beta-galactosidase. And this is a chromogenic substrate. That means a substrate, when it is converted to a product, it will be giving a particular color. Now, if the LAC-Z gene is inactivated, this gene is inactivated by insertion of a target DNA fragment into it, that means this enzyme will not be produced. So substrate will not be converted to product. The blue color will not be formed. So the development of blue color will be prevented and it gives white colored colonies. Here you can see the white colored colonies. By this way, we can differentiate the recombinant and non-recombinant colonies. If it is blue color means they are not recombinant. They are non-recombinant colonies. Okay. Now, competent host, that is the introduction of recombinant DNA into the host cell. So what is, actually I mentioned it, what competent host is. So in DNA technology, RDNA technology, the most common method is to introduce the rDNA into the living cells, that is transformation, during which the cell takes up the DNA from the surrounding environment. The DNA is a hydrophilic molecule. That means water-loving molecule. It cannot pass through cell membranes. Hence, simple chemical treatment is done with the divalent calcium ions and increases the efficiency of host cells to take up the rDNA plasmid. So, there are so many techniques to insert the DNA into the host cell. So what they do is that they will be like doing some chemical treatment here. That is using divalent Ca2 plus divalent cations. And this increase the efficiency of the host cells to take up the, they will be having the, through the cell wall pores, what will happen? They will take up the DNA. The DNA will be inserted into the host cell. That is by chemical method. Now, second method is by using heat, that is by heat shock method. The method is heat shock method. So that means the rDNA is incubated on ice at both the rDNA and the host cell. Like all the tools are incubated both on ice, and suddenly it is followed by placing them in briefly at 42 degrees Celsius. That means hot temperature. And then again putting them back on ice. It is like a shock, right? They were growing very coolly in a cold climate, not growing. They were in an incubated period and a cold temperature. Suddenly they got transferred to a hot temperature and then again back to cold temperature. It's like a shock. And this enables the bacteria to take up the recombinant DNA, the second method. Okay. Now the third method is micro-injection method. We actually discussed it, not specifically the micro-injection, that is a needle with a foreign DNA. So this micro-injection, the needle, the injection method will be like they'll be having the nucleus of the cells and they'll be using a micro glass pipette will be like using a glass micro pipette. They will be like inserting the DNA. Okay, so micro injection method means the RDNA is directly injected into the nucleus of the cells by using a glass micro pipette. So next is biolistics or gene gun. Similar to the term, it is a gene gun. And here the RDNA is being transferred to the cells, the target or the host cells. So what they do is that they'll be using some microscopic particles of gold and tungsten which are coated with the DNA of interest. So they use gold and tungsten particles and they are coated with the DNA of interest and then it is bombarded into the cells. So they are like gene gun, they are bombarded into the cells. So the gene of interest directly enters the cell's nucleus. Okay, it is gene gun method. Remember, it is the gold and tungsten microscopic particles coated with the DNA. Now, the last method uses is disarmed pathogen vectors. For example, the TI plasmid from Agrobacterium tumefaciens. So, Agrobacterium tumefaciens, pathogens of several dicot plants, is able to deliver a piece of DNA known as tDNA to transform a normal plant cell in a tumor and direct these tumor cells to produce the chemicals required by the pathogen. So by this vector, we are able to which allow to infect the cell and transfer the recombinant DNA into the host. So this is a diagram. This is a tobacco plant cell. We are using this TI plasmid, co-integrated TI plasmid, and the cell got transformed and we are culturing the cells and then we are inserting into a plantlet. Finally, we are having the cell of transgenic plants. So transgenic plants are synthesized or produced by TI plasmid. So again, we are revising the rDNA technology processes: isolation of DNA, fragmentation of DNA by restriction into nucleases, isolation of desired DNA fragment, ligation of DNA fragment into a vector, transferring the DNA into the host, culturing the host cells in a medium at large scale, and extraction of the desired product. So we need to isolate the DNA first, then fragmentation of DNA, then amplifying the gene of interest. We have to amplify it. We should make more copies of the DNA first. So this is done by polymerase chain reaction. That means it is carried out before the recombination process, and this is done by PCR, polymerase chain reaction. So here the multiple copies of the gene of interest is synthesized in vitro using two sets of primers and the enzyme DNA polymerase. So we are carrying out DNA replication inside an instrument, that is the thermocycler or the PCR machine. So we'll be adding the template DNA or the DNA of interest. Okay, then we'll be adding the primers. Then that is the primers will be needed. Then we need the DNA polymerase enzyme is needed. It is specifically Taq polymerase. Then deoxynucleotides will be added. So this will be added to an Eppendorf tube. In a small tube will be added and then it will be inserted into the PCR machine. And a particular temperature will be set, that is heat is needed for denaturation. So DNA get denatured. The primers get annealed. That means primers will be binding to the specific sites by complementary base pairing. Then the DNA polymerase enzyme and deoxyribonucleotides will be added. Added in the sense they'll be like acting there, and as a result extension takes place. So denaturation, annealing, extension will be carried out in 30 cycles, and finally we have around 1 billion copies of amplified DNA. Okay, that is PCR process. So this is about the introduction of recombinant DNA in the host cell. We already discussed it about the several methods. Now we are obtaining the foreign DNA product, that is harboring the clone DNA of interest on a small scale in the laboratory. And if any protein coding gene is expressed in a heterologous host, it is called the recombinant protein. As like the insulin protein. If you are like getting a particular protein from a particular recombinant DNA, that is recombinant protein. Now, bioreactors. Finally, we have the bioreactors. So these are like as a vessels in which the raw materials are biologically converted into specific products. And the individual enzymes are like products in the sense like it could be like individual enzymes also using microbial plant, animal or human cells in large scale. So a bioreactor provides the optimal conditions for achieving the desired product by providing optimum growth conditions like temperature, pH, substrate, salts, vitamins, oxygen, etc. So this is a bioreactor and it is usually cylindrical or with a curved base to facilitate the mixing of the reactor contents. So here you have the stirrer. The stirrer facilitates the even mixing and oxygen availability throughout the bioreactor. And here alternatively the air can be bubbled through the reactor. Sterile air is bubbled through the reactor. The bioreactor also has an agitator system, an oxygen delivery system and a form control system. Okay, so they have a motor, they have an agitator, an oxygen mixing system, delivery system, a form control system, a temperature control system, a pH control system, a sampling pot so that the small volumes of the culture can be withdrawn periodically. So as I mentioned, they have to provide optimum growth condition. Everything should be under a controlled system. So they have different control systems for each regulation procedures. So this is a bioreactor. So here, this is an example for the stirred tanked bioreactor, strange one. So increased surface area for oxygen transfer because of this rotor and bubbles are dramatically increase the oxygen transfer area. So now moving to the next term, downstream processing. The process includes the separation and purification, which are collectively referred to as downstream processing. This process is actually whatever we discussed right now is known as a downstream processing. Strict quality control testing for each product is also required in this process. Now it's time to discuss the past paper questions from NEET exam. So this question is from September 2020 and the question is the sequence that controls a copy number of the linked DNA in the vector is called. Yes, of course it is ORI site because this is a sequence which is responsible for the initiation of replication. Responsible for the initiation of replication, the ORI site. This is a specific DNA sequence within the vector or chromosome where the DNA replication begins. So option D is the correct answer. Moving to the next question, identify the wrong statement with regard to restriction enzymes. First one, they are useful in genetic engineering. This is a true statement. We have to find out the wrong one. So option A is incorrect answer. Sticky ends can be joined by DNA ligase. This is also true. We know that, right? Sticky ends are joined by the ligase enzyme. C, so B is incorrect answer. Each restriction enzyme functions by inspecting the length of a DNA sequence. This is incorrect. They are molecular scissors and they are found in the prokaryotic enzymes, which what they do, they just facilitate the manipulation of DNA molecules. They just cut the DNA at the particular sequences, that is palindromic sites. So that is correct, that they cut the DNA strands at palindromic sites. So option D is incorrect answer. C is the correct answer because it is an incorrect statement. They do not inspect the length of DNA sequence. They are particular enzymes which they are molecular scissors with particular DNA. They cut the DNA sequences. They cleave the DNA sequences. So C is the correct answer. Now moving to the next question from September 2020. Here you have to find out the specific palindromic sequence which is recognized by EcoR1. We just learned it, right? GAATTC. So here we have only one as the correct answer, option C, GAATTC as the correct one. So the correct answer is option C. So EcoR1 is a restriction endonuclease enzyme isolated from E. coli. E. coli. It recognizes and cleaves the DNA at specific palindromic sequences. And what is that sequence? GAATTC, which reads same on both strands when reading 5 to 5 prime to 3 prime direction. So when echo R1 binds to its recognition sequence, it cleaves the DNA, producing fragments that are sticky ends. So here you can see in the diagram, they are cleaving and they are forming sticky ends. Moving to next question. In gel electrophoresis, the separated DNA fragments can be visualized with the help of what? It is by the UV radiation, and we need to stain it with Ethereum bromide. So the process of visualizing separated DNA fragments is crucial in various molecular biology techniques such as agarose gel electrophoresis, where the DNA fragments are separated based on size. And here we use a stain that is Ethereum bromide followed by the exposure to UV radiation. And we can visualize the DNA fragments in agarose gel. They will be showing it as an orange colored band as well. So option D is the correct answer. So genes of interest can be selected from a genomic library by using what? So genes of interest is a desired gene and we have to select it from a genomic library. So we can use gene probes there. So what is gene probes? Gene probes are used to identify specific genes of interest within a genomic library. So genomic library is a collection of DNA fragments that represents the entire genome of an organism. And the goal is to isolate and identify a specific gene from this collection. Now, gene probe is a short labeled sequence of DNA, labeled in the sense we'll be able to identify them in under UV radiations or like under like radioactivity will be shown. So they'll be like emitting fluorescent light. So they are labeled sequences of DNA that are complementary to the sequence of the gene of interest. And when these probes are introduced into the genomic library, they will bind to the target DNA, allowing it to be identified and selected. So suppose if this is the gene of interest and we'll be adding a particular DNA probe which is complementary to the particular gene of interest and this labeled, so that means it will be emitting some kind of fluorescence light so that we can identify this is the gene of interest. Got it? Done. Moving to next question. During the process of isolation of DNA, chilled ethanol is added to precipitate what? This is an easy question. It is used to precipitate the DNA, isn't it? So during DNA isolation, ethanol is added to precipitate the DNA because DNA is insoluble in ethanol, especially in the presence of salt, that is sodium acetate. So when chilled ethanol is added, DNA comes out of the solution as visible white strands. So answer is option A. Now, next question. A foreign DNA and a plasmid cut by the same restriction endonuclease can be joined to form a recombinant plasmid by which enzyme? Yeah, the correct answer is option B, ligase. Ligase is the enzyme that joins the DNA fragments by forming phosphodiester bond. Remember this could be another question as well in NEET exam between the sugar phosphate backbones of the adjacent DNA strands. And it is essential for sealing the nicks between the foreign DNA and the plasmid, creating a recombinant plasmid. So that is they are forming, here you can see they are forming phosphodiester bond between the sugar phosphate backbones. Okay. Now, which of the following is not a component of downstream processing? That is a process what takes place in the bioreactor. Option A, preservation, expression, separation, purification. From this, we could select option B as the correct answer. Because preservation, separation, purification can be done outside the, inside a bioreactor. It is a mechanism, right, in a downstream processing. Whereas coming to expression, it is like a protein, a gene is converted, expressed into a protein. That is what expression refers to. So it is a synthesis of desired product, it could be a protein, within the host organism during upstream processing. Whereas in bioreactor, it is a downstream processing which focus on recovering, purifying and preserving the product after it is synthesized. Now, moving to next question. This is from July 2016. So, which of the following restriction enzymes produces blunt end? So, option D, EcoRV is the correct answer. So, the option A, B and C all produce sticky ends. Whereas EcoRV is a restriction enzyme that cuts the DNA at a specific site to produce the blunt end. That means they are having uniform end, no overhangings is there. Okay. Yeah. In this question, you have to identify the incorrect statement related to gel electrophoresis. So this is to find out the incorrect statement. This is from June 2024 question paper. So first one, the separated DNA fragments can be directly seen under UV radiation. This is incorrect, can't be directly seen. We need to stain it. We need to stain it with the etherium bromide and then we have to view it under the UV radiation. So option A is an incorrect statement. That means option A is the correct answer. Option B, separated DNA can be extracted from the gel piece. This is true, isn't it? By the process called elution. So option A is an incorrect answer. Option C, the fragment of DNA move towards anode. This is true. We know that DNA is negatively charged and negatively charged molecules will be moving towards the opposite charged, what is anode. So option C is a true statement, but it is an incorrect answer. Option D, sieving effect of agarose gel helps in separation of DNA fragments. This is also a true statement. That means option D is an incorrect answer. Now, moving to the next question from June 2024, recombinant DNA molecule can be created normally by cutting the vector DNA and the source DNA respectively with what? Here the answer will be option A, hint 2, because we know that to create a recombinant DNA, both the vector DNA and the source DNA need to be cut with the same restriction enzyme, so that they have complementary sticky ends or blunt ends that can be joined together. Okay. Now, moving to next question, select the restriction endonuclease enzymes whose restriction sites are present or for the tetracycline resistant gene in PBR322 cloning vector. Here the answer is option A, BAM H1 and SAL H1. So in PBR322 cloning vector, the restriction sites for BAM H1 and SAL1 are located within the tetracycline resistant gene. So actually we had seen the diagram as well in the previous slides. So moving to the next question, hint 2, always cut the DNA molecule at a particular point called the recognition sequence, and it consists of how many base pairs? We know it, right? So there's six base pairs, G, T, C, G, A, C, 6 base pairs. Now, the Ti plasmid of Agrobacterium tumefaciens stands for what? Tumor-inducing plasmid. Ti plasmid from Agrobacterium tumefaciens. Now, in gene gun method used to introduce alien DNA into host cells, microparticles of what metals are used? We discussed it. It is option D, tungsten or gold. Now, during the purification process of recombinant DNA technology, additional chilled ethanol precipitates out what? This is again repeating question. Option C, DNA. Now, the main steps in the formation of recombinant DNA are given below. You have to arrange these steps in the correct sequence. So here first will be the cutting of DNA at specific location by restriction enzyme. Then it is isolation of the desired DNA fragment. Then the amplification of the gene of interest using PCR, and finally inserting the recombinant DNA into the host cell. So the correct answer is option B. Now, which of the following is not a cloning vector? So here BAC, YAC, PBR322 are cloning vectors. Bacterial artificial chromosome, yeast artificial chromosome and plasmid PBR322 are cloning vectors. So option A is the correct answer. Now, transposons can be used during which one of the following? So here the correct answer is option A, gene silencing. Because complementary RNA of RNA interference can originate from the mobile genetic elements like transposons which replicate via on RNA intermediate. So gene silencing will be having transposons, and these transposons will be replicating via the RNA interference. So that comes to an end to the class today. Now we'll be meeting again in the next class. So bye.